we'll get started. First of all, thank you. Do we have an agenda here? All right, right. This session is meant to last about 2 hours. You probably have an agenda. So we do not have one on screen, no? Yeah, thank you. Right. So we're starting off with an introduction. Right. Yeah. Starting off with the introduction, the overview of HIV Cure remission research with different approaches with Casper Rokx. Then Jean-Daniel Lelièvre on approaches to remission. Then we've got a community perspective with Simon Collins and Memory Sachikonye. And then there's just a question and answer session.
Right. I believe I have 30 minutes. Right, so that will probably not be necessary. So we're going to have enough time for discussion. So good morning, everyone. It's a pleasure to be here together with collaborators from EATG. I was very happy to receive the invitation to speak here and to really do like a co-creation workshop on HIV Cure. My name is Casper Rokx. I work as an infectious disease specialist in the Erasmus University Medical Center in Rotterdam, the Netherlands, where I lead a clinical HIV research group focused on optimization of the HIV cascade of care and finding an HIV cure. And predominantly on that last aspect, I will focus my talk about. It is something that I hear quite often when I do my practice. People either already during their first visit with me or somewhere along the road, everyone will ask this question: can I ever get rid of it, doctor? And regardless, in my experience, regardless how good the fourth pillar, the quality of life is with people, if only for intrinsic curiosity, people ask this question: can I stop my pills? What's new out there? Do you think there will be a cure in my lifetime, in 10 years, in 5 years? Will my children live to see that? You know, can I get rid of it helps to get rid of the stigma. It is a multi-angled question in a way. So if people ask you this, it's always good to dive into that. And that's also something where a scientist should have a focus on, because that's really what drives you to find a solution for this, to tackle this problem. And I don't want to stress that I don't do this alone. Absolutely not. I've been trained by mentors in the field who gave me the opportunity and developed the skills which still is increasing and needs to increase in order to find this, because in 40 years along the pandemic, we still haven't found a cure. Well, that's completely different for something like COVID-19, where we found vaccines, etc., within a matter of 2 years. Also, at the stage where I am now in my academia, it's absolutely untrue that I do this alone. I have a fantastic team. There are 2 excellent PhD students in the back who have been with me now for several years, who really help to shape this and do the research that hopefully will bring this goal further, so that we can find an answer to this essential question in a way. Can I ever get rid of it, doctor? As an introduction, HIV is well treatable. If you treat it, it prevents death. There is, of course, the viral load that we want to get down to undetectable levels. And we do that with antiretroviral therapy, which can get the HIV viral load down in the blood where we measure it, which is a good proxy for survival. And with that, we can prevent AIDS and improve people's health. The issue with this is that HIV is never out of the body, and we can measure that. We can measure that directly in terms of measuring the virus, the reservoir, but we can also measure that by the host response. And a host response to a pathogen with immunity So what you see is residual immune activation to a certain level in almost every individual living with HIV. And this is not something to be neglected. It causes that the body ages and that comorbidities occur at a far earlier stage than in people without HIV. Unfortunately, in the far majority of people, if you stop your treatment, HIV rebounds and disease progresses. So sometimes also colleagues ask from outside the HIV field, so, well, you know, we don't hear that much anymore about HIV. It's well treatable. It's like a chronic disease. It's like diabetes, hypertension. Should we have? Why are you seeing these people in the clinic? Can't they just be with the GP on their home? Why do we need a cure? Why do we need to invest in that? And I want to make a strong point, and of course here we are all on the same page here, but this also needs to be inside the community and inside the veins of every clinic that we do need a cure. If not only for intrinsic curiosity that we should be able as human beings to solve a pathogen, it's also because there are hard endpoints that people with HIV are still amenable to health problems. And this figure shows that the life expectancy clearly has increased in people with HIV compared to people without HIV, but still is not at the same level. It really is approaching. But if you look at an individual who's 21 years old, and then look at the years that someone lives without any additional comorbidity, for someone with HIV, that's about on average 15 years. And for someone without HIV, that's about 30 years. So this really says that that you can expect additional comorbidities early on that affects lower quality of life. It's very costly for the community. There is a risk of VOC, variants of concern. We know that from COVID. There's no guarantee that will not additionally happen in HIV, that it mutates. Although the risk is, of course, low, but it is not negligible, and the stigma. So when talking about cure, and this is a bit balancing for me, I discussed this also with Brian, how deep should I dive into the pathogenesis really of cure? So please interrupt me if I need to give additional information or that I should dive deeper in it. But the essence of cure is to control an HIV reservoir cell. And as you might know, HIV integrates in CD4 T cells, which are immune cells. It stays there, and part of these cells, or small subset, goes into latency, where it stays until your therapy stops, and then it rebounds. So this is something to control either way. And these cells are hard to find. They're not everywhere in your blood. So this is Where's Wally. I think everyone knows Where's Wally. It's I think a very good metaphor to illustrate this, because I think maybe only one of you found Wally at this stage. Well, he's there. But this makes the point about the reservoir. It's findable in 1 in a million cells. These are not even 1 million people. So you need to look at the right place. Timothy Ray Brown is of course, unfortunately passed away, showed that it is possible to cure someone with HIV. And that was done through a stem cell transplantation that had a specific deletion so that HIV could not enter the body it could not enter the cell anymore. And that also signals that it is difficult, because you need another cell, you need a donor, and donors need to match. That's already rare, and that donor not only needs to match on a multitude of other factors, but also do not have this receptor on its cell surface. Second, a stem cell transplantation is dangerous. You don't want to do that without a good reason. So all people who are eligible for that have a lethal disease otherwise, like leukemia in this case. But the relevance of Timothy was not to be underestimated. It showed that it was possible to cure HIV, and several other people have been cured this way in London and Düsseldorf, and this year also by an American woman with a haplo stem cell transplantation, including cord blood cells. This is really a step in the field. Usually you give stem cells from another adult that gives that has intrinsic problems due to graft-versus-host disease. Cord blood cells have less potential of that. They're more available. They can adapt to a new host, but their engraftment as a new bone marrow is slimmer. So they did a very clever job. They found a donor cord blood cells that had this deletion and mixed it with the stem cells from a sibling, and grafted that into this woman who had leukemia after intensive induction remission therapy. And actually it engrafted enough 100 days, the stem cells of the donor cord blood, they took over. So it had a survival advantage in the setting of HIV probably. Importantly here, no single HIV reservoir cell was found in 100 million blood cells. So to think about the Wally, right, 100 million is a lot, but is not even the amount of cells we have in the body. Talking about cure, either we should increase the immunity to tackle the virus, or do something to reduce the reservoir. And traditionally it has been as a sterilizing or a functional cure. Well, what's the difference? Sterilizing means there's no virus in the body. And this is by usual therapies like now, it's almost impossible to reach. And it's very rare if you have an immune system that can reach that on its own. But only two ways to at the moment to reach that goal is a stem cell transplant, as I just explained. But you could also use gene therapy. And this is very early on, I think, that we are with gene therapy. But basically, you could do two things. One is you took a large blood volume from a person with HIV, and you genetically manipulate the CD4 T cells to not have the entrance receptor anymore, and then you reinfuse that in an individual. So that's really an autologous infusion of functional immune cells to HIV infection. The other way would be to use gene therapy to excise the integrated HIV in our own genome. Problems here are the yield of the first approach, and second is, of course, off-target effects. If you're going to manipulate your gene, your DNA, there can be off-target effects in other genes that you do not want to mess with, and then you can have oncogenic changes, which of course is a very unwanted side effect. So this is really early on, but at least the stem cell transplantation shows it's possible. The other one, the functional cure, is there's still virus left in the body, but we accept that because either it's in a very low amount, it's inactive, or it's controllable by our own immunity. And you can reach that by maybe passive immunization, where you optimize your T cells, again, ex vivo, you manipulate them so they become more potent towards attacking HIV that's in the body. You call that CAR T cells, or you use broad neutralizing antibodies that recognize a broad range of HIV antigens that might be expressed on cells. The other thing is active immunization. You activate your immune system to produce these antibodies, and that's something you can do with therapeutic vaccination. It's either focused to induce these antibodies or to induce potent anti-HIV T cells, which is not a sinecure to do. Because of course with having HIV, you already have an active immunization ongoing. So you need to do better than biology, basically. And the third aspect is shock and kill, where something I do, where I'm involved in my research, and I'll go on later to explain that a little bit more. So what are the challenges? Remember, HIV reservoir is findable in 1 in a million. So you should look at the right place. Even if you look in a significant, so if this is all the blood of a person's body, like 4 to 5 liters, even if you look in a significant proportion, say 1 liter, but the Wally is not there, you will not find it. So you need an assay that is sensitive, and you need a large amount of blood to look at it in order to say something about the reservoir. 3, discriminatory power. These 2 red guys are reservoir cells, but one is dead and the other one is standing. With some of the assays, we can't distinguish between that, but it is relevant. 4th, there might be other reservoir sites, and the question mark is now is hidden behind the video. But if you don't look at it, if you don't sample it in those those areas, you can't, just don't know. 5, when is low enough? I said that low amounts are probably important to get to functional cure, but what's low enough? 6, a biomarker would be really nice. A biomarker that's everywhere in the blood, easily measurable, and that is very well correlated to that one wally or the absent wally. Switching to our last part of my presentation about the Cure research line. I was asked to say something about that. What are we doing also together with the EATG? So there are 2 priorities. One is to understand HIV Cure reservoir hurdles. Second, find interventions, intervene. Focus areas here are which drugs to use. We should personalize approaches early on. We should focus and not forget understudied populations that are not and involve them early on. And it should be done in a network. You need to synergize intellect together. The times of Galileo Galilei, where he alone developed the mathematical modeling, the physics, and the experimental setup to prove that gravity is there, or is probably behind us. So I'm fortunate to work in the Erasmus MC HIV eradication group. I was one of the initiators there, and we collaborate with the HIV Vereniging, which is the Dutch EATG, so to speak, with people from biochemistry, hardcore biochemists who dive deep into how does the genome work, right? The HIV integrates in the gene, it acts like a gene, so we need to know how are the genetic and epigenetic factors there. With people from virus sciences, where one of my mentors, who unfortunately passed away, Professor Dr. Charles Boucher, worked and led the department. He was an inspiration to this. He was the inspiration to start this over 10 years ago, and I was fortunate enough to be involved in it. We have people there from pharmacology and from immunology to try to cover all aspects here. And what we basically do, I thought about this, how can I best depict it? Basically what we want is a cure drug pipeline. Everything starts in the laboratory, where we do unbiased and targeted screenings of potential compounds, trying to purge the reservoir, come to a cure, and characterize host responses with it. That goes into this tunnel, and promising cure drugs, preclinical, can be identified by them. And then you want to process them through this pipeline, including in the internal medicine department where I work, where we do the translational work together with the collaborators in order to prove that a certain drug that comes out of this pipeline is able to target the reservoir and improve the care improve the health of people with HIV. And one way to improve this is to really get the personalization already into the drug pipeline. Probably, as we know with all drugs, so that with cure drugs is saying people respond differently to different drugs. And that's also why you have variation in responses, as I just showed you in that graph. So it's probably important to stratify and study potential cure drugs, stratified to key characteristics of people that usually influence responses to therapy, like sex, therapy duration, what's your disease stage, HIV subtypes of viral and host factors. First do that in the lab, and then do a clinical challenge to see if that really correlates well. And in order then to identify the most promising drug, to further translate into randomized clinical trials in a personalized, predictive way, hopefully. Eventually, you need to study this in a trial. This is like a general template for that, where you found an optimized regimen, maybe 2, you randomize people to that. You measure the HIV reservoir. At one point, you probably need to do a treatment interruption. That's the proof of cure. But one way to make this more efficient is by keeping involving this cure drug pipeline, where continuously new regimens, drugs are being identified, and then you can add that to this trial platform, and that's called an adaptive trial platform. So you make more efficient use of both your preclinical work and the trial. So my last section is on the European consortium EU Cure, together with EATG, borderless innovations to cure HIV, with stakeholders from throughout Europe unified in one group. A focus area is understudied population, and we try to start a research project on HIV subtypes. The most studied HIV subtype in cure is subtype B, and this is the subtype that's most prevalent in Western countries, but not in the rest of the world, where 90% of the population with HIV has a non-B subtype, which is now almost completely unstudied in HIV cure research. So that seems like a very relevant topic to dive into, especially because these viral genetics differ between subtypes. They behave differently, and this also affects clinical outcomes if you look at antiretroviral therapy. So probably also in terms of cure, a difference can be expected, and we need to tackle this early on. At least that's our opinion. Already a lot has been done. This is just to show how many studies have been done in this group, and this also shows that it's very complementary and a lot of potential to cross-link and cross-fertilize. This is just one example. These are 4 key studies on shock and kill cure drug strategies from different groups that included an analytical treatment interruption. And by just eyeballing it, you can see that some individuals in each of the group have a prolonged time to viral rebound, but their numbers are small. And you can think if we pool them, then you increase the number of people, and you might be able to find a signal there instead of just looking at these studies alone. So there's a critical mass of knowledge together. This allows meta-analysis on larger cohorts, and also with the groups that we have, new trials on the most promising compounds early on. So with that, I would like to end. I believe I'm in time. And we're saying that more research on HIV cure is necessary, is something we do together. That's why we're here, we're working towards effective clinical strategies. And especially, this is also a call out from me to you, if you know of a way to can help our studies on HIV cure, especially with non-B subtypes, that would be very helpful. It's really, not a challenge, but it's really important to study this group. But it is hard to find people from just sitting in your hospital. So if you, from another different angle, know ways to aid with that and are willing to, that would be very welcome. You can reach out to me or to one of my PhD students, Katie Hensley. And I would like to stop by thanking all the collaborators and colleagues without whom I will not be stand here in the funders. Thank you very much. I don't bite.
Maybe I'll start off with you. Just one comment on there. One of the things that strikes me about when you're running through the choices between sterilizing and functional. Functional seems like the more manageable option rather than sterilizing. And when I'm looking through that list, it's good to see that Erasmus is a joint consortium, because I think it has been the case that so many people have been working in silos over a long period of time that we don't seem to have any coordination of what's going on. And one of the things that struck me was, I remember going to, I think it was Melbourne 2014, but it was a World AIDS conference where there was quite a big session on therapeutic vaccines, and you mentioned them there. they seem to have completely gone off the boil. There was quite a lot of interest in therapeutic vaccines for a while. Do you know why that is, or is it just that it was deemed to be perhaps even an option for long-term treatment, not just the cure? So do we know why there isn't as much research into therapeutic vaccines now, or do we just not hear about it?
We will hear about just after about the therapeutic vaccine.
I didn't. I heard your comment, of course, but I didn't hear the person online. What is it?
Oh, it's going to be in the next presentation.
Yes.
All right, I'll not start then, and all the answers will come there, I believe, on vaccination. I don't get them.
Does anybody else have any other comments or questions on that? Actually, maybe I do have a question. Where do you think you see the most, it's actually worrying, actually, can I say, just to begin with, when we look at that and we think that where all the subtype activity is in, obviously, where all of the good treatment is anyway, in terms of the world. So where do you think there's the most chance of actually getting concrete results through within the next 10 years? Sorry.
Yeah, I will look in my glass ball. All right, we know where to go to, right? We know it should either be the end point is either an ELISE controller or a post-treatment controller. The difference between them is that the first is a very engaged immune system in order to control your HIV. The second is a person where the HIV is very latent and present in only very small amounts. So those are two phenotypes that you want to reach. It's easier to say what I think will not be done on a large scale at that stage. I think stem cell transplantation will remain very hard. I also think that gene therapy is not ready for the clinic. So that said, I do think that the combination of targeted activation of the immune system, either passively or actively, together with improving antigen presentation in the reservoir cells, is the way to go in order to get a more durable control over HIV, at least from a perspective what's most reachable. I think what we need to do, and that's also why I think the personalization is important, is to know in which people should we look for the success of this strategy first. Probably that needs to be in people where the HIV reservoir can be reactivated quite well and where there's still a preserved immune system to start with. And that's, I think, people either with an acute, we've started treatment in acute HIV infection. But that's a glass ball prediction, so don't give me to it.
Well, again, thank you very much for that. Unless anybody has any other questions or comments. I must admit, I know that later on we've got Simon in memory talking about what the community thoughts on the cure. But I'm glad to see that kind of research going on, because having lived with HIV for 84 years, so since 1984, sorry, for 84 years, since 1984, I know that we go through periods where there are new magic drugs that come along that are absolutely wonderful. And of course, a magic drug is only a magic drug for a few years, and then you find out what all of the associations are. So, and I do think that when we see the accumulation of other issues going on with long-term living with HIV, I am very glad to see this research carrying on. We're now moving on to our next speaker, but I don't know how to get that all organized. So Jean-Daniel, we are moving over to you now, and you're apparently going to do one of my answer one of my questions on therapeutic vaccines, so that's very good. So thank you very much, Jean-Daniel. It's over to you now. Thank you.
Okay, do you hear me?
Yes.
Perfect. So I will try once again to share my screen. So the idea here is to give you a feedback on the implementation of a clinical trial, a therapeutic HIV vaccine trial, EVA-TO2. And I've entered my presentation from scientific expectation to reality on the ground. These therapeutic HIV vaccine trial was carried out through the EVA consortium, and this consortium includes 39 partners from 11 countries in Europe, 4 in Africa and the US, with leading scientists and community experts in the fields of both molecular and structural biology, vectors, adjuvant delivery, immunology, clinical science, and biostatistics. And we need all this kind of expertise to build our trials. So the consortium was launched 6 years ago in Paris, and the objective of this consortium was to set up both HIV prophylactic and therapeutic trials and cure trials, but also to try to develop new vaccine, both for the prophylactic and the therapeutic vaccine strategies. So we want to build new trials, but also new vaccine to be used in these trials. So as mentioned by the previous speakers, there is two ways to see the cure strategy. The first one is to reach eradication of the virus, and there are only few models for this. Timothy Brown was the first one, and to date we only have 4 or 5 new patients that could reach this kind of eradication. It's quite different with functional cure, and here the elite controllers are the model for this kind of cure, and they represent 0.5% of HIV infected patients. It is now quite widely accepted that this kind of lifelong remission, functional cure, is a more realistic objective than sterilizing cure, and it's important also to emphasize that it's also easier to implement and maybe safer. And it's also important to mention that both type of cure strategies approach need to combine a different strategy, and for functional cure you need to combine therapeutic vaccination and immune-based therapies. As previously mentioned, treatment interruption is usually associated with a rapid viral rebound approximately 2 months after treatment interruption, but it could be more rapid. However, in some patients, and as mentioned also previously, the post-treatment controllers, you have, after treatment interruption, a control of viral replication. And it's also the case in elite controller, but elite controllers control the viral load without any kind of previous treatment. So all these elite control are consistently associated with T-cell control, and we have a lot of publication that confirm this statement. If you look at what happened, the relationship between the viral replication and CD8 T-cell response during HIV infection, you can see that in people living with HIV that did not currently receive treatment, you have high viral load, and this high viral load induces a CD8 T-cell response. Using treatment, you will decrease the viral load, and as a consequence, you will see a decrease in CD8 T cells. So what we want to do using therapeutic vaccination is to mimic what is currently seen in elite controllers, meaning that you have here low viral replication, in some of them undetectable viral replication, and it's correlated with a high and important CD8 T cell response. It's also important to mention, and it's indicated in this slide, that you have both during HIV infection, a quantitative defect of the CD8 T cell response, at least after treatment, but you also have a qualitative impairment of the CD4 T cell response, which is not the case for elite controllers. So for some combination therapy used with the vaccine in functional cure strategy, you will either increase the number of your cells and for other, increase the quality of your T cell response. So the goal of the therapeutic vaccination is to induce a CD8 T cell response, but the induction of such a response requires endogenous protein expression, which was previously achievable with live attenuated vaccine. And this, we know that this kind of vaccine can be used in people living with HIV. So a different platform have been developed to allow presentation of HIV epitope to CD8 through vaccination. And currently, you know, better this kind of platform because they are widely used for at least two of them for COVID-19 vaccine. So you have the viral vectors, you have the RNA vaccine, and you have the DNA vaccine. And for each, the principle is the same and consists in integrating a nucleic acid fragment coding for the protein of interest in the vaccine platform. As you can see here, you see that it's the spike because it's an example with the vaccine against the SARS-CoV-2, but you can integrate it either in the DNA, for instance, of an adenoviral vector, or in the RNA of mRNA vaccine, or here in the DNA of a DNA vaccine. So you also need to choose what part of the HIV genome you want to include in this kind of vaccine. And the sequences coding for GAG, POL, and NEF are the ones generally chosen because CD8 T cell response, again, these proteins are associated with viral load control, as you can see here during HIV infection, but mainly and especially in elite controllers. And you have in those patients a very important response against the GAG protein. And once again, we want to mimic with the therapeutic vaccination what happened in those particular patients. Finally, the one also important thing to consider when looking at therapeutic vaccination is the use of heterologous vaccination strategies, meaning the combined use of different vaccine platforms, but expressing the same level of the same viral antigen, and the combination can increase the T cell response. Here you have an example. This is not results from a therapeutic vaccine trial, but a prophylactic one. But at the end, the immunological effect is the same. You can see here we are looking at a particular parameter which is called ELISpot gamma interferon. It's the production of one cytokine by the cell after CD8 T cell after vaccination. And you can see here the level of induction of this kind of response, and it's I think quite clear in this figure. If you use only a DNA vaccine or only a recombinant adenoviral vector, you can see that the impact is not as effective as the use of the combination of the two vaccines. So what were the choice of vaccine that we aim to use in our clinical trials? We use a combination of two vaccines, one viral vector, the MVA-HIV-B vaccine, and one DNA vaccine, the GTU HIV-B vaccine. They both encode for the same kind of protein, GAG, POL, NEF. and from a subtype B vaccine, because we aim to perform this clinical trial mainly in Europe. So we choose a subtype B virus for the choice of the sequence we want to use in our vaccine. And immunogenicity and safety of this vaccine have been studied for the DNA vaccine both in HIV patients and in healthy volunteers, and for the MVA HIV-B to date only in one prophylactic vaccine. However, we were also able to look at the safety and immunogenicity of the combination of the two vaccines in the RNAs-VR1 clinical trial we have performed in France. So we thought that the combination was the best one to use to date, at least in Europe. So we decided to design a first version of our clinical trials in 2017, the EVA-T01 trials. And in this trial, the objective was to compare the efficacy of the combination of these two vaccines. And you have here the design of this clinical trial with the placebo arm and a vaccine arm, and you have the phase of immunization followed by 24 weeks phase of treatment interruption. As mentioned previously, combination of strategies is quite important, and you have to combine therapeutic vaccination with different kind of immunomodulator that could impact different part of the immune system, or, as mentioned, increase the quality of the quantity of the CD8 T cell response. As an example, you can see here the impact of one immunomodulator and one vaccine strategy in a non-human primate model, and it's a study performed by Dan Baruch in New York. You can see here the impact in terms of viral replication after treatment interruption in those monkeys, and here in black you have the viral replication without any kind of intervention. You have here the impact of one immunomodulator, and here it's an agonist of the TLR7 receptor, and with a very poor effect of this immunomodulator. The effect of the vaccine seems to be greater, but more importantly, you can see that you have a synergistic effect of the combination of both here, the vaccine and the immunomodulator. So we decide for our clinical trial to use vedolizumab for as a combination with our two vaccine. Vedolizumab is an antibody, monoclonal antibody directed against one integrin called alpha-4 beta-7, and it's currently used for ulcerative colitis and Crohn's disease. So we know quite well the safety, some issues that can be associated with the use of this kind of treatment. So it's known to be quite safe in regular use. And this vedolizumab is not to impact the trafficking of CD4 T cells, mostly in the gut. And you can see here the impact on this particular structure present in the gut, and they are clearly decreased after treatment with vedolizumab, and it could therefore have quite a synergistic action with the vaccine. And also there are a first study performed in the HIV macaque showing that the use of the infusion of this monoclonal antibody in the HIV infected animals allow to control viral rebound after treatment interruption. However, these quite interesting results were not confirmed in the three further macaque study. In the meantime, however, studies have been performed in human with the use of vedolizumab alone, and you have two clinical trials, one performed by an American team and the other by a Canadian team. The first one include 18 patients, and the viral rebound and the effect of the treatment was compared not to placebo but to an historical control group coming from a previous therapeutic vaccine trial. Unfortunately, you can see that there is no clear effect, at least on the endpoint, was the ability to suppress viral load and below 400 copies after treatment interruption. However, if you look carefully at all the individual results, you can see that 30% of the patients reach 26 of treatment interruption, week 26 of treatment interruption without meeting criteria to restart ART, meaning without more than 100 copies of a viral load for more than 4 weeks. And also here you have the results from the Canadian study who involved a smaller group of patients because there were only 8 patients. However, they did not also use a control placebo arm, but they were able to show that there is a difference in terms of viral control after treatment interruption if you use a higher dose of vedolizumab. So we decide to include vedolizumab in combination with our vaccine, and we choose the number of infusion performed in the Canadian study, the higher dose, the same kind of criteria that used by the Canadian team, and I show you why we use these kind of criteria for restart ART and not the very restrictive criteria used by the American team, which did not allow to have a viral replication of more than 1,000 copies. So we decided 1 year later to launch this new clinical trial, and we add vedolizumab with the vaccine in our clinical trial. So you have the design of the new vaccine that include the previous vaccination with 3 shots of vaccine and 7, a little more, a number of infusion with vedolizumab. However, we face a first issue, which was the bankruptcy of the company that produces our DNA vaccine. So we decide to use only the MVA vaccine and not the combination. And finally, it was fine for us for 2 reasons. First of all, because we were able to obtain very good efficacy in terms of induction of immune response only with the MVA vaccine. And as we are not in a clinical trial with only vaccine, but with a combination of vaccine of vedolizumab, it seems okay to use only one vaccine and an heterologous combination of vaccine. So I will go now in details in the use of the different criteria for viral to restart ART. We use these kind of criteria, and especially we allow in our clinical trials to have a quite important viral replication after treatment interruption, which was not the case in the American study. And why did we decide to do so? Because we have some result from our previous vaccine clinical trial performed in France, the ANAS-VRI-02 Lite, which was a randomized clinical trial performed in different centers in France, a phase 2 clinical trial that include 105 patients, 50 who receive a vaccine combination and 34 who received the placebo, and it was followed by a treatment interruption period of 12 weeks. And in this clinical trial, the results show that if you met the inclusion criteria, that quite important, and you need to have a CD4 nadir of more than 350 cells per ml and a CD40 cell count at baseline of more than 500 baseline. There was no individual risk during prolonged ATI of even 12 weeks of treatment interruption, even if the viral load rose very high, and in the median of viral replication, both in the placebo group and the vaccine group, was quite high in our clinical trial. However, we face one issue in this clinical trial related to a treatment interruption, and it's not directly for the patient, but for the relative, because we unfortunately saw HIV infection transmission during the ATI period. So we think it's quite important to inform people involved in ATI clinical study that they should be very careful about these risks, and that their partner could have, for instance, access to PrEP. So why was it quite important to allow this very high replication? Because if you want to compare clearly the efficacy of your vaccine and to compare it what happened in the placebo group, the most important parameter is finally the viral set point. And to have the viral set point, you need to allow replication for a long time, because there is no correlation between the set point and the slope, for instance, of viral replication. And we were able to look at all these kind of parameters using results from different clinical trials performed either in France or through collaboration in the US, and we look at more results for more than 248 patients. So at the end, the most important parameter was the analysis of the area under cover of viral replication after treatment interruption. So we decided to launch this new version of our clinical trial. However, as is the case for all of us, we face the issue of the occurrence of the COVID-19, and it was clearly shown that there is some risk to perform ATI clinical trials during the SARS-CoV-2 pandemics. And here you have an overview of a quite interesting paper published 2 years ago, and discussing all the issues to perform such kind of trial during the SARS-CoV-2 pandemic. So what are the risks, and how we deal with those risks in our clinical trial? The first one was epidemiological risk related to the incidence of infection. It's quite difficult, obviously, to control, and we decide to include at the end only patients that have completed a full COVID-19 vaccination. You have risk related to the state of health, and as mentioned by the previous speaker, unfortunately, there are a lot of other comorbidities associated with HIV infection. So we set up a new validation committee to be sure that we will not include people with comorbidities that put them at risk for a severe COVID-19 infection. And there is a risk related to the trial itself. It seems that there is no clear specific issue related to the use of MVA or vedolizumab. There is some risk related to ATI, but there is no clear answer related to this specific risk right now, and that there is related to the constraint of the trial and the frequent visit to the hospital, and it was quite difficult to deal with this risk because it's quite difficult to, for instance, conduct sampling outside the hospital at the time of ATI. At the end, we all agree to discontinue trial treatments at the ATI if patients become SARS-CoV-2 positive during the trial. So we decide to launch wide. It seems that the situation with the COVID-19 pandemics was safer and people have been vaccinated. However, we face new issues. The first one was the monkeypox infection. Indeed, the MVA, the viral vector used in our trial, is also the vaccine used to vaccinate against monkeypox. Even if the MVA used in this context come from a different strains, our MVA and the MVA used by Bavarian Nordic and present in Invanex or Jynneos vaccine are slightly different. However, vaccination with MVA HIV-B that we have used in some previous clinical trials and we aim to use in EVA-TO2 are able to induce both a response against the HIV proteins, including the vaccine, but also response against monkeypox. Some data performed through our consortium in France have shown that a minimum delay of 15 days between two of injection of MVA is quite necessary to avoid a deleterious impact on immune response. So what could be the impact in our specific clinical trial? First of all, can we consider that vaccination with MVA HIV-B protects against monkeypox? While we don't have any kind of marketing authorization in this indication. And if it's not the case, it will be necessary to synchronize vaccination between the two, which could be quite difficult. And finally, the main issue was difficulties facing in rotating patients in the clinical trial. At the end, the target number of population was 69. Unfortunately, 4 months after the start of the trial, only 3 patients could be included, and perspective on the whole center was a maximum of 24 people. While the number of people living with HIV in the different clinical centers was more than 20,000. So we decided to stop the trial as there were also time constraints associated with the trial related to the loss of the apparent fundings and the expiration of the vaccine. We don't know yet what are all the motives and the reason why the people don't want to be included in our clinical trial. However, we have a still ongoing study looking at motivation and experience related to the refusal of participation, and it's performed by an INSERT team here in France. So the interest in trials and studying cure strategy in the course of HIV infection remains quite high, as also previously mentioned, and both for you and for the scientific community, as shown by the great number of ongoing trials. However, the implementation of at least of large phase 2 clinical trial remains complicated, even with strategies that have shown their safety. And it will be crucial to understand the reason that led to the failure of EVA2 in order to be able to consider more efficiently the conduct of other trials in the future. So I would like to thank all the people involved in this clinical study, mainly Song Ding from the Eurovac consortium and Sheena McCormack and his team from the UK. I would also, like my colleague, want to pay tribute to Professor Charles Boucher, which was a marvelous person and a marvelous scientist, and who was involved in our consortium. Thanks a lot.
Thank you very much, Jean-Daniel. Again, do we have any questions or comments that people would like to come up to the microphone and make here? Nobody? No? I suppose I would like to make one comment, and again, I'm feeling that I'm showing my age here, because you're talking about elite controllers.
Mm-hmm.
And there was quite a lot of research done into elite controllers in the early years, I would say. But how easy is that now? Because after the START trial, we really weren't waiting. You know, we didn't have CD4 cutoffs before you started therapy. So I would have thought that so many of the people that might have gone on to become elite controllers actually went on to antiretroviral treatment anyway. So how easy is it to study elite controllers now?
Yeah, I think that there is two types of control. You are not speaking of elite controllers, but rather of post-treatment controllers, and the immune correlates could be quite different. And however, at the end, it seems that to be either elite controllers or post-treatment controllers, you need to express to our current knowledge quite specific receptor linked to immune response that are called class 1 HLA. And for the elite control, the main actors seems to be the CD8 T cells, while for the post-treatment controllers, it seems that could be either CD8 T cells, but also another type of cells that are the NK cells. So there are still a very interesting research on the treatment after ATI, the natural control of viral replication after ATI. And there is some research that are currently performed, at least in France, with the Riviera Consortium, to try to understand who will be the people that will control viral replication after treatment interruption. However, while I explain you that you may have CD8 T cells or either NK cells, maybe you may have also other actors involved. So it will be quite important to go on and to try to see what happened and to decipher the mechanism involved in the control viral replication after treatment interruption, and it could arrive either after treatment during primary infection, but also obviously it's less frequent, but also during chronic infection.
Yeah. Any other questions or comments? I would just say one thing. It is so unfortunate the trial comes up against not just one virus, coronavirus, but also along comes monkeypox, just to make it a bit of a party too. But I think maybe the lesson for all of us, and we'll maybe discuss this at a later stage, is trying to look at maybe the hesitancy and why people maybe don't feel that they want to get involved in the medical trial and the cure. So that's something for all of us to look at too.
If I may, one comment on the SARS-CoV-2 pandemic, and it was mentioned by my colleague that we were able to obtain quite rapidly a vaccine of SARS-CoV against SARS-CoV-2, and it was quite efficient. But we have to keep in mind not us, but other people and the policymaker, for instance, that if we were able to do so and to have this kind very rapidly this kind of vaccine, it was because we performed a lot of studies with HIV vaccine, and all the knowledge on the viral vector, on the DNA vaccine, on the mRNA vaccine, on the use of heterologous prime boost strategy come from the HIV field. So it's important to thank you all and to thank us all, and it was due to what we have performed in all these years that we were able to have this kind of vaccine.
Well, thank you very much, and I hope you can hold on for a little while longer. Thank you, Jean-Michel. Jean Daniel, sorry, Jean Daniel.
No problem.
Yeah, I think that's somebody else I'm thinking about. But at the moment, we're now moving on to two presentations, the community perspective. We're starting off with Simon Collins and then going on to memories. So Simon, if you could come up first, please. Thanks.
So just next. Yeah.
So fantastic. Nice to see everyone here. There's a few more chairs if people want to come forward.
Is that working?
Yeah. So I didn't really prepare slides, but I've got a few just to sort of hang a talk on, really, and just to give some informal comments from a community's perspective about cure research overall, and some of these pointers were put together by Sean. I'm not sure Sean's here. Yeah. And so over the last 10 or 15 years, there's been a huge interest in cure-related research. We have to be very careful with language. We mustn't just call this cure research because it ends up with people enrolling in studies that they think they're going to be cured. No matter how much you write in informed consent that this is not a cure study, if you call it a cure study, someone will say, ah, yes, but it might just be me, I might be lucky. And so you have a disconnect, it's not actually ethical. And it's very difficult to overcome because even if you write cure-related, I'm sure people will still hope it will be them and they might be cured. But over this interest in cure-related research coming after such amazing successes with treatment has also been funded, and that's the important side. Is a lot of US funding went specifically into cure-related research, and the researchers that are working in this field are also working collaboratively on an international basis. So the researchers in the US will be including researchers from Australia and the UK and European countries and some African countries. But as one of the earlier slides shows, there's a big gap in actually where the studies are being run and who they're being run. And so I'm going to include a little bit about that. I'm going to make 3 or 4 points. The first one is that there's a fantastic piece of work by the IAS, the International AIDS Society. Every 5 years, they do a review of what's happened in the previous 5 years and a sort of roadmap for the scientific questions and ethical questions that we would ideally like answered in the next 5 years. So partly the reason I'm here, I haven't done a huge amount with cure research, but I was on this group that helped put together the 3rd of these reports. It was published on World AIDS Day last year, and it's really worth reading. It's published in a high impact journal called Nature Medicine. And if you start off there, it's probably pretty tough reading. Complicated, technical, condensed. It summarizes, since the previous report, so this is the 3rd of the 5-year reports, it probably puts in 170 new studies. That gives you an idea of how dynamic this field is and how it is moving on and how we are finding better ways to understand what's involved in it. The second version here is it was put together by IAS as a briefing for journalists. It's slightly less technical. So it's okay. It's translated, I think, into Spanish, Portuguese, and French. A little bit more information there. And the third one, there were maybe a group of about 10 or 20 community advocates or activists that were involved in producing this main paper from all continents, really useful group. This is, did we get the handouts? Great, okay, so there's some handouts. So this was something that was put together by the community people that were involved in the main journal, and it's a really easy, non-technical summary of saying what we thought were the most important issues from the community, and also what the most, in an easy language to understand what this technical version is all about. So for most of us, I'd start off with here, I'd work backwards to the journalist one, and then print off the main paper and go through it slowly, just to understand some of the technical terms, because it's written in really concise language. It's just highly technical. And if you try and work through and understand the language involved in the research, it will make it easier to follow the research and where we're going. So one of the easy things to explain from that very geeky technical report is that the viral reservoir gets talked about, the applause to go down, gets talked about as this tiny reservoir, and it's true that you have to, in somebody, there was a fantastic paper actually, and it was somebody who started treatment in really early infection, maybe before they seroconverted in the US, and they were on treatment for 10 years. And as a part of research, they looked really hard to find out where HIV was, whether it still existed. And there's also one of these slides here, and they found HIV 1 in 2 million cells. So it's tiny, it's not there. This person, against doctor's advice, decided to, I think it was David Ho's lab he was being seen in, decided to stop treatment, just out of curiosity. Could this be enough? Could I be cured? And even with that low reservoir, viral load rebounded. And so there's this impression that the viral reservoir is tiny, but actually this is the viral reservoir. 10 times 10 to the 10 or something. That's like, there's better, like 10 million or 10 billion cells. So HIV is there in significant quantities, and this is one of the graphics, the illustrations that's from the main paper, and it breaks it down in terms of cell type and where in the body this might be. We might have cellular reservoir in lung tissue or in fat tissue. And so it's a really tricky thing. Eradication is difficult. When we're coming back to language, I don't think you'll find any person that wants to refer to this as sterilizing the cure. Horrible. You jump back, this is not a nice sounding thing. Find better words, find more friendly words to talk about it. But this is an easy way to explain how complicated it is to cure HIV. And you can slowly work through different types of cells, where they are in your body, where they are, and just how large it might be. So some of the community report, that some of our input into this technical paper included, we want everyone to know about cure-related research. So anybody who's diagnosed HIV positive, living with HIV, in any country in the world, no matter how much they know about treatment or want to know about treatment, they really at the back of their mind want to know whether they'll be cured. So one of the things we were saying to the IAS is, great, you're doing this geeky paper, but don't forget simple language and really translate, produce stuff that's easy to translate. And then wherever you are in the world, you can quickly read where we are with cure-related research. And that can be optimistic. A lot of us don't have easy lives, don't have straightforward lives. Our health maybe isn't at the top of the list of the things we're worried about. We're worried about jobs, where we live, employment, heating bills. So the fact that there's all this research going into cure research can be optimistic. it can boost you and it can make us a smaller sort of community. And we can share the language, we can share the research. And so please translate some of the stuff that's going on if your main language is English. And if you're someone like the IAS, please remember that in terms of producing material easy to understand. We also said we want research to be inclusive of sexuality, sex, gender, region of the world where it's been where it's taking place, age, all these sorts of things. Age, we don't want age caps on cure-related research when many of us are past that age anyway. We still might be interested in contributing. I'll talk a little bit about inclusion criteria in a minute, and to also include social research. So if you're spending this money doing clinical research, ask the people that are taking part what their experience is of your study. Ask them what you think a cure involves. You know, for some people, it's just not taking treatment. That's the main thing that they upset about and that affects their day-to-day life. So you could overcome that goal by just producing better treatment that maybe only involves an injection every 6 months. You're sort of or injection every year. You're sort of getting close to a lifestyle that would be similar to being cured if you only had to go to the doctor once a year. And so we're at a very exciting time where treatment research is overlapping with cure-related research. And there's another point there, which is that if you're on an experimental treatment where you stop treatment, we'll talk about ATIs in a bit, you're vulnerable to when that viral load comes back. And so another overlap between cure-related research and research in general is that we want to have a point of care home test for viral load. You can't wait a month even to find out if your viral load is still undetectable, if you're on a cure-related study, because by that time, if your viral load's rebounded, it will be 20,000 copies or 50,000 copies. So other things we said, sorry I have to read from the slides, the disconnect between ethics. So people still enrolled in cure-related studies thinking they're going to be cured. And they sign a paper that says, I will not be cured. There's a disconnect between ethics. It's always been there. Global research, we want all regions involved in performing this research. We want expertise in labs in different countries where the research at the moment is very limited. We want community preferred language, no sterilizing cures, and we want to have extended follow-up of these cases. So where we're talking about post-treatment controllers, does everybody understand what that means? Okay, so we're in a really, most people do, but I bet a few people don't, because post-treatment controllers are people who've been on treatment, they interrupt treatment, and then their viral load stays undetectable. So the Visconti cohort was published, this got a high level of interest in international reports about HIV and HIV development, but they didn't report, amazing researchers, and they found this, and they reported that maybe 1 in 10 people, French study, could be a post-treatment controller. And it comes up with the questions that Brian was asking, you know, are lots of us on treatment who don't need to be? Well, it was, that's not the case. Most of us definitely need to be on treatment. If you stop treatment, your viral load will rebound within a few weeks. But it took a long time to have follow-up on those people published and presented. So community activists were saying to the researchers of the Visconti cohort, great you found this, but we want to know in next year whether these people are still controllers, and the year after, and the year after. Don't leave things hanging, because the science could change, and we might be making our decisions based on inaccurate information. So if you look at I'm going to go forward actually. So TAG have done a fantastic project, and they went back tracking studies, cure-related studies since 2018, and they looked at the baseline demographics. Who was included in this research? And it was overwhelmingly men, it was overwhelmingly white men. And they went back through the registry and they just it's Richard Jeffries thing, really. And now he's just pulled together a report, and this is what you can see in terms of the over 4,000 people enrolled, and less than 20% are women. And hardly anyone's non-binary, and then there's missing data. So some studies are publishing data about cure-related research. They don't even put to gender and sex and age in them. This is about geographical region and ethnicity, and you can see that the balance as well is very skewed. So we're only studying a certain group of people in something that has relevance to everyone. And so if I go back 2 slides, I should have done this in a different order. So there's some very exciting data coming through that there's differences based on sex, and that this is, I mean, I'd say to researchers, you should be using your initial studies in animal models, and monkeys should be using male and female monkeys and macaques and things, and see what's going on. So there might be a dynamic here that's very different, and the implications for cure-related research for women may be very different to the implications for men. It might be a different target. And we've known this for decades, that viral load is generally lower with women. And actually that resulted in women starting treatment later, based on when they had viral load control criteria, which was not appropriate. The requirement for need back then would have been earlier treatment. So Sean wanted this group here to talk about the personal impact of cure-related research on participants. And I think that's important. And we just heard about the EVA2 study, and I was on the call that decided that recognised that the study couldn't meet its planned enrolment. It just wouldn't be powered to show anything at all. And so the question for the EVA study was it ethical to continue enrolling a few people into a very difficult study if you knew you wouldn't be getting good data out of it? And so it was on that basis that we thought it's not fair to continue to put people, maybe even if they're committed to the study, when at the end, when the study finishes, you're not going to get good data because there's not enough people enrolled. And so it was difficult being on that call where the EVA2 study's been planned and for over 6 years. Unfortunately, it was better just to say something might come out of it, but not in its current format. So these studies are difficult to be a participant in. And really, I'm going to say something now, which maybe I shouldn't say, is that the current standard of care for treatment is 1 pill a day. And it's a pretty small pill. And it's tolerable, and it will normalize your life expectancy as much as any treatment can. If you're complicated and have a difficult history, then maybe your life expectancy isn't equal to being HIV negative. That's the caveat that guidelines say life expectancy is normalized, but that's based on data for people that have never injected drugs and they've never had hepatitis C co-infection. And actually our lives are more complicated than that. So life expectancy is knocked a bit, but you're never going to find a treatment that's better than a single pill fixed dose combination, so long as you can tolerate it. And if you can't, switch to another one you can tolerate. And so anybody's quality of life will probably go down during the period they're in the study. And that has to be clear. And there's financial implications if you have to do 6 visits and take time off work and travel to the venue. This is quite difficult. It's only a small number of people are able to participate in studies. There's issues about you might go through all this and you're in the placebo arm. So there's a study running at the moment in the UK called RIO. It uses 2 long-acting monoclonal antibodies, and you get those infusions maybe once every 6 months. It involves a treatment interruption, but there have been cases where people have done this, they've interrupted treatment and stayed off therapy for over a year with no viral rebound. Now that gets pretty close to what lots of people want from a cure. There's no guarantee this would happen in everyone, but it's a really exciting example of where the cure-related research overlaps with treatment-related research. You know, and these antibodies actually both been bought by, they were developed at Rockefeller, they've both been bought by Gilead. In terms of access, the person who developed these antibodies has told me that they could be produced for $200. $200 with the right economies of scale. And if that's a single shot and looks after you for a year, that's manageable as a cure intervention in any country. So it's not just, you know, geeky things that can only be done in wealthy countries or high-income countries. Cure-related research is important wherever you live in the world, and it's probably more important if you're in a low-income country that's dependent on US funding largely for your treatment. You do not want to be worried about who the next US president is. And I'm quoting Moses Supercharger, who says this is why in Africa we want to have a cure, because we're able to know, you know, who the next President Trump might be. And if you suddenly decide to cancel our drugs, or if we're living in a country where the reality is we have stockouts. You know, so if you're in a country where you have access to a single pill once a day, you're doing okay. You probably don't have a huge need for a cure. But if you're not, and that's the majority of people in the world, that's the majority of the 28 million people on treatment, don't have stability in terms of access to treatment, alternative treatments. You know, if 1 3-in-1 doesn't work for me, I'll change to another. And that's not an option most people have. And the ATI side of this is that if we've got undetectable viral load, and we've been undetectable for several years, that's our standard of care. That's what we're used to. In our head, that's how we think. We're undetectable. We don't need to go to the doctor even very often. We have our blood tests, and that's it. We don't, like, you take a treatment interruption, and interesting data coming out of the Rio study in people who've done this because they believe in research, they want to know if there'll be a cure, they'll go through a difficult process, is they have actually a fantastic time not taking 1 pill a day. I don't know if it's been measured, but it is measurable. You know, when I was on twice a day, I didn't think treatment could get any better. I thought once a day doesn't make any difference to me. It changed my life. But also, when I was on 3 times a day, I thought that was fantastic. I can manage this, thank you very much. I'm very well organized. I didn't think twice a day would make a difference. It changed my life. People who take treatment interruptions, analytical treatment interruptions, they will have their life changed. They suddenly are not connected to the drugs at all. And then they have to restart. And I know that's difficult. And so the bit that we sort of forget is you'll go into research for all the nice reasons, you'll have a response, and then people have to go back on treatment again. And I think people are finding that quite difficult. So it's sort of a be careful what you wish for. You know, I advocate for cure-related research. I think we'll get there, you know, in whatever the 10 years is, we'll get a cure. Science would be amazing. But we want to know, we also be a bit more careful, make sure people going into studies recognize that they might be surprised at how good they find time off treatment and how difficult they might find it going back on the treatment again afterwards. And so that's a few things to talk about when we have discussion. And so actually these complicated cures, these complicated studies like EVA were sort of acceptable before COVID. We're in a different world, you know, and it's a challenge to research now is to design things that maybe don't involve 7 studies, having infusion at a clinical centre and all the travel and all the cost and the difficulties there. Maybe you could redesign studies where maybe the researcher comes to your home and gives you the infusion. It doesn't increase your risk of anything from travel. So that's thank you.
Thanks. I don't know if anybody has any quick questions for Simon there. Can't see any. So Memory, would you like to come up? Thank you very much.
Yes.
Hi, everybody. I was asked to talk about what cure would mean for me personally. So my name is Memory from the UK CAB and HIV IBIS. So I was diagnosed with HIV 20 years ago with a CD4 count of less than 6 and a very high viral load, taking up to 18 tablets a day. I'm now 56 and my HIV is now undetectable. My viral load is undetectable and I'm taking 3 tablets a day for my HIV. I have other conditions, which is a chronic kidney disease. I've now been 7 years post-transplant, and I still have to take my immunosuppressants and other tablets. So that's a total of 8 tablets a day, 7 in the morning and 1 at night. So my current quality of life is generally quite good. So what are the pros if I were to be offered treatment or if I were to get treatment? It could be a relief from daily pill taking and the polypharmacy, especially that I'm also aging. There's that psychological impact on the possible lifestyle change. No more daily medication, but for me it doesn't apply because I still have to take my immunosuppressants. It would mean less clinic visits to the HIV clinic, but I still have to go to my renal clinic often. On the whole, it could also help with countries that have stockouts if people didn't have to worry about having to take treatment, knowing that they are cured. Would this help combat stigma? That's still questionable. I find that there's a lot of cons in cure research. I mean, as you have all heard, the process is very complex. It's very difficult for anyone to go through. Our current treatment, as someone just mentioned, is very effective and it's easy to take. If cure becomes available, and there's the question of whether it will be available and accessible and affordable to those who actually need it. Stigma and discriminatory attitudes will always be there. HIV will not go away. And the promise of cure gives false hopes to some people. I think as someone mentioned that already, I won't go through that. And might affect those who are, might affect adherence of those on treatment. And there's always the worry that you still get reinfected. And is this rollout likely to happen in our lifetime? I don't think so. So clearly, the cons outweigh the pros for me. We're all aware of what U equals U can do. I find it liberating. I don't worry about passing on the virus to anyone, be it a partner, be it a mother who's going to have a child. And I would like us to focus more on ensuring that everyone globally who needs treatments has access to it. There's new exciting science about long-acting treatments. That, if I were to be given an injection once every 6 months, that's what that's more appealing to me than every 2 months. So it would be, it would work better for me. I just want to acknowledge my friends who I also asked, and they completely agree with what I had to say. It was just something that we're all aging and all getting menopausal, lots of other problems, but we are okay the way we are. Thank you.
Thank you very much. Does anybody have any questions to ask of Memory there? No? Well, we're just about to go into a wider Q&A discussion, but if I could just say one thing here quite personally about it. I'm fascinated and I really love the idea of a cure, but I must admit the thing that would scare me about participating in a trial. I'm not naive, I'm not thinking I would necessarily be the one that would benefit from it, but when I first started on HIV therapy in 1995 and then went into kind of drug trials like ritonavir and Sachikonye, which was a wonderful trial, we didn't even have viral load tests. We had CD4 tests and viral loads came along. And for me, one of the defining factors in my life has been getting to that stage when I didn't have a viral load of 1 million and 400,000 and things like that, that was undetectable. And I suppose for me, one of the greatest fears of when I look at some of these trials, and something that needs to be addressed for a lot of people, I would think, is actually just seeing that, viral load just going up a little bit and it being defined as okay because the CD4 count's still the same which we did here earlier on. I would have concerns about that personally. And it isn't just the U equals U thing, that is very important. But it's actually, for me, if you like, that comfort blanket of since the turn of the century, the year 2020, having had an undetectable viral load. And for me, it feels like I wouldn't say a badge of honor, but it's certainly huge comfort. But maybe if we could have a wider discussion, a Q&A on some of the presentations we've seen today and what you maybe think some of the issues are in cure research. And thanks to all the speakers we've had here today so far, and also, by the way, thanks to Sean Hussein, who helped to put this together as well. But if we could move into a Q&A, would somebody else like to come up and talk? Yes, thank you.
It's not like a question, but after thinking for all the speakers, it was a great presentation. But I just wanted to say, as someone living with HIV since 2016, I think I'm feeling a little bit lucky because I was diagnosed in a time that treatment was amazing. I didn't even have any symptoms, and when I immediately started on treatment, I was undetectable almost immediately. And since then, I never had any problems with my 1 pill a day. And I only recently needed to change it because I needed to take ADHD pills, so it was just interaction. That was the only reason for me to change. So, and I'm working in sexual health and HIV support. So I'm talking to so many people living with HIV. And when I'm talking to them, because if they are recently diagnosed in the era of ART, it is really important to go to the research. Personally, I committed to the COVID researches and the monkeypox researches myself. But when it comes to HIV, do I really want to disturb my 1 pill a day, just waking up in the morning, taking my pill, 0.01 millisecond and move on with my life? Because also it is not communicated clearly to me why those trials are important and what the phases are. And I am not I'm in love with science, but I don't know the science terminology. I'm not a doctor. But when I'm looking at the studies, I would love to know more about what the phases, what kind of stages it is going through, and then understand it better, and then maybe commit to a trial. So as a patient perspective, that's why I am having hesitancies to join an HIV trial right now. And I think my colleagues are also, because they're happy with 1 pill a day and they don't know where HIV is coming from 20 years ago, 25 years ago. So they don't really want to go there and disturb their quality of life. So I think this should be considered as a...
Thank you very much. Any other comments there? I see your point, I must admit, but Simon made a very good point, because I do remember being on, three sets. In fact, that's Sachikonye and ritonavir. I can't remember how many pills it was, but it was a massive number of pills. And I think it's maybe easy to get to the stage where you think I'm in a good place. I'm on 1 pill, one pill once a day kind of thing. But at the end of the day, I also look with envy at the new long-actings coming through and thinking, oh, if only I could get to the stage where I had an injection once every 6 months, going to the clinic, getting some blood tests. So, but the other interesting point that Simon made there, I think, was he talked about, in a way, the overlap between long-acting treatments and cure coming in. So I think that's a fascinating part. Would somebody else like to come up and say something? Yes, Ben.
Hi, I'm Ben. I don't want to be all gloom and doom at all. I thought it was fascinating. Actually, I've been part of these kind of cure things since the 90s, and it's really interesting to see how things have developed and they haven't developed. And some of the underlying points were, it's still absolutely shocking that that tag showed that 20% only of the study participants were women. And I think we have to really own that as us. I mean, that's really part of our problem. And the other thing I really appreciated Simon bringing up again was the absolute importance of changing our viral load test system. And we are so behind in pushing diagnostics change. You know, many people don't have access to viral load tests at all. And we pointed out what that problem means is that people can't really imagine where they're actually at. They have some, if they're lucky, they have a CD4 test, but what does that tell them? So it's just, I guess it's a certain amount of humility about the problems that we've actually still sort of accepted in our own movement, in our own health system. And HIV is one of the best funded systems in the world, and we still have these long-lasting problems. And as Simon pointed out, you know, if we're not actually testing female, my terminology may get female monkeys, you know, that there may be real substance to this difference between women and men that really is holding back certain kinds of research. So that's my own comment.
Thank you, Ben. I might like to add to that as well. I think part of the comment that Ben makes there, and Simon's made it too, is that effectively, in a way, we've lost a sense of emergency and urgency. Because when we saw COVID coming around, all of a sudden things that we'd been asking to change for a long time could be changed because there was no other option. And I think we should maybe learn from that. And especially when you talk about diagnostics, and you know, mea culpa here, because I'm now chair of the Partners in Science program at the EATG. I only took over last December. And I think it's at least, I think it would be at least 2015 since we last had a specific ECAB on diagnostics. And that clearly is one area where we could be moving, you know, much further and much faster than we currently are. It's just that there doesn't seem to be the, if you like, the push. And if there isn't the push, then the push has to come from us. It's that simple. So, someone else like to speak? There must be some people here who would like to speak. Sorry, Casper, could you have to come up here? Sorry, thanks. Doesn't get recorded otherwise.
Right. So I think it's very relevant to hear the community opinion on cure, right? That's why scientists like myself and the people from the field should come together, because obviously if you're in your ivory towers, you know, to use a metaphor, you can think that cure is get rid of the virus, right? That's the end point. But here clearly, and I think the point is very well made, is that the current treatments is going into a direction that can be felt as a cure as well. So the question from me to you here would be, so what would a cure feel like? So not what is it, but what would it feel like? Would it feel as a cure if you just indeed have an injection every 6 months or yearly, but then you do home testing with a viral load? There can be a discrepancy in how a cure is defined from different perspectives, and some people might not find testing themselves every week or every month as a cure, but it might be that many of us think that is something that you would be happy with and you would feel cured. So I'm just wondering, raise this. Would that feel like a cure, or is that not a cure? Is that not the cure we want?
Would anybody like to answer that? Yes.
Well, I personally refuse to switch over to injectables as treatment. The reason for that is because I'm lazy. And as a patient, I don't like to go to the hospitals every 2 months to get the injectables, see my doctor, talk to the blood test or whatever. I don't want to do that. And if a cure requires me to engage with the healthcare setting or just going or getting checked is good, but you know, going to the blood test or going through testings again, or just if there is a hesitancy, if there's a little bit doubt in there, then I wouldn't step into that circle, I think. that would be my only thing. But if it's like you said, if I need to get an injection, if I have a definitive answer for that, if you're going to give me an injection for a year, and then you're going to tell me that a year later, okay, you are HIV free right now, go over in the wild and run away, I would do that. But if I need to be constantly for the rest of my life going to get checked and going to this question in my mind, is it okay now? Am I cured now? Or is it going to come back? Then I would rather take the treatment forever and be you equals you and then be safe. That's my personal thought.
Got another person coming up here to speak. Thank you.
Hi everyone. Not really prepared to speak today because I didn't know I was going to be involved in any discussions, but I just wanted to add that concerning treatment and maybe vaccine uptake or being involved in clinical trials, I would say the approach will not be like a one-size-fits-all approach. Participation in trials and maybe treatments will be determined by different dynamics of different groups and communities and cultures. There's some cultures that will not uptake vaccines just because of their culture and their understanding of maybe the science behind the treatment and the vaccines is not being explained to them properly. And one thing I'd like to mention is coming from my background. So there's a religious and faith-based issues concerning uptake of treatment generally. So that is one of the reasons why in some of these hard-to-reach communities, there's poor response to uptake of services generally, because some of the faith-based messages that are being passed on to groups are really maybe not correct, I would say, because they are being passed down to members by people who are not very experienced or who don't have the science knowledge. So I think for cure and treatment, faith leaders would also need to be part of like public health messages. They would need to be maybe invited to discussions because they are the ones that the community trusts. The community really, the religious people really trust their leaders, and they will take any message that the leaders pass on to them. And so I think they would be important people in discussions around uptake of treatments and vaccines and being involved in clinical trials generally. Stop here.
Any other questions or comments before we, as we're getting towards the end of the session? I would maybe slightly like to answer your question, Cass, because you asked, would it feel like a cure and what would I think? I must admit I'm slightly at odds with you there on that one, because I think clearly, an injection once every 2 months is not where we would want to be with long-acting antiretroviral. But if I could get to the stage where you give me an injection every 6 months or every single year, it would feel like a cure, and it would mean I could go about my daily life just so different. So I wouldn't need to think about packing drugs to go on holiday. I wouldn't need to think about where I am at any given time. So, but I think the comment that was just made there is that we're not all going to be the same. People are going to be different. People have different levels of comfort and different levels of needs. So I think it's more a case of trying to engage with as wide a community as you can to get responses to this. We've got a reasonable mix of people in here today. We haven't heard from anybody from pharma yet, by the way, and I noticed there are a few from their end, so if they have any comments to make, that would be interesting. Do you have a comment to make? Yeah, up you come.
So thanks, Brian. And well, just one word. I mean, I came halfway through, and I'm very sad that I missed the first part of it. So thank you very much. For me, it was an eye-opener. I think the fact that there is no one-size-fits-all is very, very clear. It's also nice to see that we are kind of the right path to get to what you guys need, that you have all the options available To what Casper said, please keep on shaking the average hour, because I think that's how we all community look into different sides of it. We get things done. So yeah, but it was an eye opener, and thank you very much for that.
Thanks, Felipe. So while we just think about getting towards the end and summing up, I think there are a few take-home messages here, and they've been made by some people here, and it's all about perhaps us having to take a bit more action as well. Simon made the comment about how people need to know about the cure. People need to actually know on a far wider scale than they currently do about what the cure is and what it means. And I think the other thing there was the language thing, because Simon did talk about sterilizing cure. We've got to come up with something different for that one. That really doesn't sound very good at all. Yeah, it's not. But also the. Yes. Yes. That sounds like tonight's last installment with a woman as a doctor to be Doctor Who, sorry for people who aren't familiar with the television program. Yes, exterminate, eradicate. But before we actually, one of the other things I will take home from this is that it's highlighted to me, from both Simon and Ben, that we need to be doing more on diagnostics, because our diagnostics are almost as old as Methuselah, let's be perfectly honest, and that must be because we haven't done anything to try and get them changed. We haven't really done anything to try and get home delivery on diagnostics and stuff like that. So that's a course of action that I think the EATG has to take on board in the community in general. So while we get round to the finish, first of all, I'd like to say thank you for everyone for coming here today. Been a very good turnout, quite a wide variety of people in here. Very glad that you came along on the opening day of the conference. I'm really glad to have you all back here. I come from Edinburgh. This is my Glasgow, just along the road from me, and it's just so wonderful to be at a conference with human beings, seeing them, touching them again. Thank you very much. So I'll just like to thank the speakers. We had Casper Rokx, Jean-Daniel Lelièvre, not Jean-Michel, thank you, Jean-Daniel Lelièvre. We've had Simon Collins and Memory as well. Thank you as ever, thank you. And thank you for everybody for making their comments. And I'm sure they'll be able to fill out, where is Giorgio? They'll be able to fill out an evaluation, presumably?
No.
No?
We are inviting, if you want to continue following us on our activities, and there will be a report released on this meeting, so please use the QR code here on the screen, and you, at the end of the page, you can register yourself for updates. Thank you.
Thank you. And as I say, one last thank you as well to Sean Bassane, who's one of our Canadian members who couldn't actually make it here, but who put this whole show together. So thank you very much, everyone. Thank you.
Thank you.
Thank you to you.